video we'll be doing an overview of communicable diseases. So there are a few things that you need to know in this chapter, which is what are communicable diseases, looking at 4 specific pathogens and the types of diseases that they cause, a little bit more about how the human body actually defends itself against infections, and also if you're doing separates, there's a little bit more about the microbiology and plant diseases and defense that you need to know. So let's start with the fundamental stuff, which is the general overview of what is health and what's communicable diseases. So generally speaking, health is focusing on the physical and mental well-being of a person. Now, in biology, we'll be mainly focusing on the physical aspect of it, and in this chapter, we'll be looking at something called communicable diseases. So communicable diseases are mainly infectious diseases. That's what the communicable aspect is. So these diseases are actually caused by something called pathogens. Now, pathogens are any microorganisms that can cause an illness. Now, there are 4 pathogens that you need to know. So these are the 4 pathogens that you need to know, which are viruses, bacteria, fungi, and protists. Now, in particular, you need to know a bit more about viruses and bacteria in the sense of how they actually make us ill and how they reproduce. So let's start with viruses. So the first thing to note is that viruses are actually can reproduce inside our cells. Viruses on their own cannot actually reproduce, unlike most of the other living things that we've got. They have to actually land on a cell, which we call the host cell, and they will inject their own genetic material inside the host cell, which then combines with the host cell's own DNA. And when the host cell is actually making proteins using the DNA, it will then also produce the viral proteins as well, assembling all of those viruses. So effectively, it makes the host cell into a virus-making machine or factory, and once there's enough viruses inside, it will burst from the inside out, releasing all of that to the environment. So that is also the reason why viral diseases cannot be cured, because you're gonna have to destroy the host cell in order to get to the viruses, which is what the viral diseases actually do in the first place. So that's why we can't cure viral diseases, but we can actually give them perhaps treatments to like painkillers to kind of sort out the symptoms. So there are some animal and plant viral diseases that you need to be aware of. So for animal disease, there will be measles and AIDS. So you will learn more about the details of it when you actually do go through this chapter. But measles is one of the most obvious symptoms will be a rash and a fever. That's especially obvious in children, and that's why most people actually will get a measles vaccine as a newborn child. AIDS actually stands for acquired immunodeficiency syndrome, where the virus actually attacks our white blood cells and stop your immune system from working. So more on that when you actually go through the details of the chapter. So for plant diseases will be a tobacco mosaic virus. So as the name implies, it mainly infects tobacco plants, and it produces a mosaic discoloration pattern on their leaves. That's why the name tobacco mosaic virus. Now on to bacteria. So bacteria is a type of prokaryotic cell. So they are able to reproduce rapidly by something called binary fission. So we've learned previously that animal and plant cells reproduce by mitosis, where the cell splits into 2 through a series of steps. Whereas for bacteria, it's quite similar. It's still about a cell splitting into 2, but the process happens a lot quicker at about once every 20 minutes. So bacteria's ability to reproduce so quickly leads to why it is able to infect individuals quickly as well. One of the major ways that bacteria make us ill is by releasing toxins. So these toxins are basically chemicals that are able to interact with the cells, either stopping them from working or perhaps stopping a certain enzyme from working, and that is why it makes us sick. Now the good thing though is bacteria can be killed by antibiotics. So this is a medicine that specifically targets bacteria and it kills them by breaking their cell wall, for example, or interacting with the way that they reproduce. So that you will learn more about antibiotics in the next chapter. So salmonella food poisoning is one of the animal bacterial diseases that you need to know. The symptoms would include things like diarrhea, vomiting, stomach ache, fever, etc. The thing is though, This one is usually caused by the bacteria inside raw poultry like chicken and ducks, or even raw eggs sometimes. It's not likely to kill people. It's not a very fatal disease. That's why when someone gets salmonella food poisoning, they don't normally get antibiotics, because you don't want to have bacteria getting antibiotic resistance. So it's something that you just simply let it pass. And the types of question you get asked is usually, how do you prevent the spread of salmonella, which is basically focusing on having good hygiene, or good maintaining the hygiene in the kitchen, washing your hands, etc. Gonorrhea is the other example of bacterial diseases in animals. Now, this one is an STD, which is a sexually transmitted disease. So typical symptoms would be pain on urination, green or yellow discharge from the genital areas. And in a really serious situation, if it's left untreated, it could lead to infertility or ectopic pregnancies, which is when you got the embryo implanting itself into the oviduct instead of the uterus, which is very, very painful. So this is something that can have a really serious, long-lasting effect. So therefore, if someone does get infected by gonorrhea, they will be treated with antibiotics. So the third pathogen is fungi or fungi. You don't need to actually know how they make us sick, but the key thing you need to know is the disease that they cause, which is rose black spot. So as the name implies, they target roses, and they would produce black spots on the leaves, which means that it's destroying the chloroplast. Hence they have less photosynthesis, less glucose made, stunted growth, and etc. So that's fungi. Similarly for protist, which is the fourth pathogen you need to know, you don't need to know how they actually make you sick, but the key type of questions that come up will be the disease that they cause, which is malaria. Now, malaria is a disease that tends to be asked in an exam question. Not so much in terms of asking about symptoms or treatment, but it's more focusing on data interpretation and thinking about how do you prevent the spread of malaria. Now, malaria is a disease that is caused by protists, but is actually transmitted by mosquitoes. Now, we use the word vector to describe mosquitoes. So vector is the thing or the animal or the object that actually causes the transmission, but they don't necessarily get sick themselves. So what happens is that the protists will reproduce inside the mosquitoes as well as inside the human blood cells, like red blood cells and liver cells. And then actually, when the mosquito lands on someone with malaria, the protist enters the mosquitoes, reproduces inside them, causing variation, and when the mosquito go and suck blood of a healthy person, that transmits the protist to the healthy person, and that's how they are transferred. So the typical questions will be things like, they will ask you to suggest and explain one method of reducing the spread of malaria. So it will be mostly focusing on how do you reduce the number of mosquitoes or preventing mosquitoes from biting people, basically. So things like mosquito nets, remove standing water, improving the education, so letting people know how to prevent malaria spread, etc. So these are some of the ways that you could mention in the exam questions. So these are the 4 pathogens that you need to know, and like I said, you specifically need to remember viruses and bacteria a bit more. Now that we have looked at a bit more about in terms of the diseases there, so there are some general things that you can mention when it comes to suggesting things to prevent the spread of disease. First of all is personal hygiene, so making sure you cover your nose when you sneeze or cough and wash your hands regularly, especially after you've done that. Those are some of the basic things that can prevent the spread of disease. You can also talk about how can we destroy certain vectors. So for example, mosquitoes, as we mentioned in malaria, or sometimes mice or rats in particular, that they are also typical vectors that could cause the spread of diseases. You can also mention the use of antiseptics, which are chemicals that can destroy pathogens on the surfaces of stuff. So like, for example, an alcohol spray or kitchen spray. So those are examples of antiseptics. You could also mention quarantine if it's a very serious disease. You can quarantine someone or isolate someone while they're infected and make sure that they don't spread the disease any further. And finally, you can also mention vaccination, which is about injecting dead or inactive pathogens into the body, triggering our own body to generate and make those memory cells. For more details on that one, that will be covered in the next chapter. So you are required to know a bit more about this one. So keep an eye out for a bit more information on vaccination in the future. Now, moving on to human defense. So when thinking about infections, we need to also be able to describe and explain how the human body actually do two things. First of all, is to prevent the entry of pathogens in the first place. And secondly, if the pathogen did enter the body, how can we defeat it and kill the pathogens before they make us sick? So we can largely classify human defense mechanisms into two parts, which is non-specific versus specific. So for non-specific responses, what we mean by that is that it doesn't matter what pathogen enters the body is still defeating them in the same way, right, regardless of what it is. So first one is the skin, which acts as a barrier, a physical barrier to stop the entry of pathogens. Another thing that you can also mention is sebum, which is a layer of oil on the surface of the skin that traps the pathogens and stops them from going any further. You can also mention mucus and cilia. So on the inside of our trachea and digestive duct or reproductive ducts, they will be cells that actually release mucus, which is a sticky substance that stick dust or any pathogens in the stuff that goes through your body. And what happens is once they got stuck in there, the cilia are finger-like projections that can waft and move in a synchronized movement to move the mucus out of the body. So when you have phlegm in your throat, that is what the mucus is. And the reason why you can cough the phlegm out is because the cilia is moving the mucus along with the bacteria or pathogens out of your body, and so that you can cough it out. Another thing that can also happen to the phlegm is that you can swallow it as well at the top of your throat, and it will go into the stomach where the hydrochloric acid will be there, and the acid will be able to destroy the pathogens, obviously, because of the acidity. So there could be other things that you mentioned, like lysosomes and lysozymes in your tears or your eyelashes, etc. But these are the four key ones that you might want to mention if you get asked an exam, especially the skin being a barrier and the mucus and cilia. That comes up quite a bit. Now, as for the specific immune response, that is mainly involving white blood cells. Now, what you need to know is that they are able to do three specific things when infected by pathogens. First of all, they can do something called phagocytosis, which is a process where the white blood cells can change its shape and engulf the pathogen. So for example, if here is a white blood cell, and if there is a bacteria that is approaching or nearby, what happens is that the white blood cell then changes its shape, kind of like Pac-Man a little bit, and wraps the outside of it around the pathogen. And eventually they are going to engulf, we use the word engulf to describe this process. They're going to engulf the pathogen so that the pathogen is inside the white blood cell, and eventually it is broken down and destroyed and digested. So that is the process of phagocytosis. Another thing that happens is that white blood cells can produce antibodies. Now, this is where it's important to know the difference between antigen and antibodies. So we've got two cells here. We've got the pathogen on the left and a body cell on the right. Now, the way that the white blood cells tell which one's our own cell and which one's a foreign cell is by the cell surface protein. So we've got the cell surface protein here. Now, those proteins are called antigens. Now, we've got two different types of antigens here. So the one on the left, where the pathogen is, we've got a pointy antigen, and we call it, let's say, the foreign antigen because it is a foreign cell. Whereas the one on the right of our body cell, we call it a self antigen because it's one of our own body cells. So the way that it works is that the white blood cells are able to produce antibodies. Now, there will be loads of different types of antibodies. So for example, these are two types of antibodies. So these are small proteins that are able to recognize the antigen in order to tell if it's a foreign or a self antigen. So for example, in this case, we've got this antigen here that is made that has got a complementary shape to the foreign antigen. So it's able to bind to it and recognize it as a foreign cell, as a pathogen, and therefore it will then attract the white blood cells to go and kill it. So for example, it can attract more white blood cells to do more phagocytosis, engulfing the cell here. So that's the difference between antibodies and antigen. So antigen is a cell surface protein that allows cell recognition. Antibodies are the proteins that bind to the antigen to recognize it as a foreign cell or not. So the final thing that white blood cells can also do is to produce antitoxins. Now keep in mind, like we mentioned earlier, one of the pathogens that can produce toxins would be bacteria. So let's say if this was a bacterial cell, they're able to also produce tiny toxins that goes off and interact with how our cells work. So the way that the antitoxins would work is that the antitoxins can then bind to them. So it stops them from binding to the other parts of the body. So therefore, it's not going to affect how the body works. So it stops what we call, it neutralizes the toxins. Right? So neutralizes the toxins. So that's the phrase that we use. So those are the 3 things that white blood cells can do specifically to kill the pathogens in our body. Now, if you're doing combined sciences, this is all you need to know for this chapter, which is what are communicable diseases, the 4 pathogens and how they, and some of the diseases that they cause. You will need to know a little bit more detail, obviously, about how you treat them, what are the symptoms, how do you prevent the spread. But generally speaking, this is what you need to be able to kind of explain a bit more in detail for the exams. However, if you're doing separate sciences, then there are 2 bits that you need to know a bit more about, which is microbiology and plant disease and defense. And so keep watching and then we'll go into that now. So the first thing that we're going to look at is microbiology. So microbiology is a way of saying that, or looking at practicals that helps us study how pathogens like bacteria would work. And one thing that we tend to do is we want to know how to grow the bacteria in the lab and how do we investigate and do experiments around that. So while we're plating bacteria or dealing with any sort of microbiological experiments, we need to pay attention to something called aseptic techniques. Now, aseptic techniques are anything that we do in a lab while working with microbiology to reduce the risk of contamination. Now, inoculation is a word that is used to refer to the growing of bacteria. So when you're transferring bacteria onto a plate to grow it, we call it inoculation. So it's really important that we try not to contaminate the sample with anything else, because that would obviously interfere with the accuracy of the experiment, or it would just kill the bacteria that we're trying to grow, which is not ideal. So there are some examples of aseptic techniques that you need to be aware of. So for example, there are 3 here that we're going to have a think about. So first of all, you always would have a Bunsen flame in the area, and the Bunsen flame serves the purpose of having this convection current. So any bacteria that is in the air around it is going to be, because the air is going to be heated up near the Bunsen flame. So when the air is heated up, the density decreases, so they go up. And as they go up, that means you're actively bringing any bacteria in the area away from the sample. So you want to work near the Bunsen flame and not away from it. And actually what happens is the bacteria with the hot air is going to rise, but when it cools down, it's going to sink back down. So if you're working away from the Bunsen flame, you're actually going to receive those contaminating bacteria that you don't want in your sample. So it's very important to work near the Bunsen flame. You will also put it as a blue flame because that's the hotter bit, so that's why it's better that way. The other thing that you also want to think about is to limit the number of times you open the lid and also the degree of opening. So rather than removing the lid entirely and then work with the plate, you want to just open it slightly and just slide in whatever material you want in the gap rather than opening the lid. So that again is about preventing the entry of pathogens landing onto the surface of the plate. A third example would be sterilizing equipment before and after use. So for example, this is what we call an inoculating loop. So we put the loop bit into, let's say, a bottle of bacterial solution in order to transfer it. But we want basically heat up the loop before using it so that it kills any pathogen that's on the surface of that, and then wait for it to cool down before dipping it into the bacterial solution and then spreading it, and again sterilizing it in the blue flame after use so that you don't have any bacteria staying there or like going everywhere in the workbench. So these are some of the examples of aseptic techniques. Other things would also include like sterilizing the workbench before and after use, washing your hands before and after, doing the practical, or wearing gloves and changing gloves regularly. So there are many things that you could say, but these are some of the main specific ones that are quite unique to working with microbiology. Then let's have a think about how do we interpret certain results, or what would it look like, and what are sort of the stuff that you need to know. So let's have a look at this one. This is what an agar plate would sort of look like once it's covered with bacteria. The yellow bit is signifying when we've got a whole agar plate with nutrient agar along with the bacteria that is already in there. So one of the things that you need to know is actually a required practical in this case, which is to investigate the effect of different antiseptics on the growth of E. coli, which is a bacteria. So what you will do is you will put 4 paper discs. You can see that there are 4 discs there. And what you will do is that you probably soak those discs in different antiseptics. So what would be a good thing to do is, for example, you have a control sometimes to see, let's say it's just water or just the paper disc, to ensure that the paper disc itself is not killing the bacteria. But let's say for this example, we're going to be putting antiseptic A, B, C, and D into the 4 different areas of the plate. And what you then do is that you incubate it for a bit. So what we mean by incubation is that you're going to put it in a warm area at about 25 degrees Celsius or less. So that could be about for 24 hours. That would be good enough time for it to work. And then you observe the plate afterwards, which may look something like this. So there you have it. This is an example of what it would look like. Now what you'll see is that there are some clear areas around those paper disc, and that's to be expected because the antiseptic would diffuse out of the paper disc and kill the bacteria in the surrounding areas. So then you got this clear sort of circle. Now that circle is what we call zone of inhibition, where there is no bacterial growth. So that means that in this case, antiseptic B, which is this one here, is the most effective antiseptic because it produced the largest zone of inhibition. So the types of question you get asked is, which is asking you to interpret the plate or to calculate the area of zone of inhibition, which would be pi r squared. Now it's really important that you make sure that your answer is comparative, so saying things like it's bigger or biggest or the largest, and this one's the smaller or smallest. So for example, B would be the most effective antiseptic because it made the largest zone of inhibition, whereas antiseptic C is the least effective one because it has the smallest zone of inhibition. So that's what you need to be able to interpret in an exam question. So the required practical in this chapter will be mainly focusing on aseptic techniques and explaining some of the aseptic techniques that you need to know, and then be able to interpret and explain how this practical works. So finally, let's think about another separate only content, which is plant diseases and defense. So there are a few ones that you already know from the previous bit, which is the pathogens that causes them. So you've got rose black spot, which is caused by a fungus, and also you've got tobacco mosaic virus. Another thing that they also like to ask you is mineral deficiency. While this is not a communicable disease, it does belong to one example of a plant disease. That's why they put it here as well. So there are 2 minerals in particular that they tend to ask, which would be magnesium deficiency and nitrate deficiency. So magnesium is the ion that actually makes chlorophyll. So if you see a plant that is lacking in magnesium, they would have yellow leaves. And nitrate is a key component into making amino acids, which then goes on to make proteins. So therefore, if you see a plant with nitrate deficiency, they usually have what we call stunted growth, which means that the plant is smaller than what it's supposed to be. So the typical things that they like to ask about, plant diseases is how those sort of conditions affect the plant and explain why it would affect it. And it's mainly about how much photosynthesis that could occur. So this could be due to, let's say, they have magnesium deficiency, therefore there's less chlorophyll, less light absorbed, less photosynthesis, less glucose, less proteins are made, and therefore stunted growth. Or it could be rose black spots or tobacco mosaic virus, where they destroy the chloroplast, hence less light absorbed, less photosynthesis, less glucose, less proteins, therefore stunted growth. Or like I said, nitrate deficiency directly, less amino acids, less proteins, therefore stunted growth. So ultimately it always leads back to stunted growth, but you need to be able to explain that sequence of events. Which leads us to plant defense, which is the three main mechanisms that the plant uses to defend itself against any pest or pathogens. So we've got physical defense, which are mainly focusing on individual specific structures that the plant cells would have. So for example, the existence of a cell wall or the lignin cell wall will be a physical defense. A second way of defense will be a mechanical type of defense. So these will be things like, if the leaves have fur or like a small hairs on them, or perhaps they have the waxy cuticle, or perhaps they have spikes on them. So those are mechanical defenses as well. Then the last one will be a chemical defenses, which, as the name implies, when the plant is releasing certain chemicals that either kill the thing that is destroying its own cells, or perhaps it could be certain chemicals that when herbivores or pests eat the leaves, it will actually give off a really bad taste or bad smell, so it deters them from eating them anymore. Another typical one that they like to ask is mimicry as a way of plant defense. So that probably would be classified as a mechanical defense. So the type of questions that they like to ask will be, they will show you a picture and describe how the plant is defending itself, and then they will ask you to name the type of plant defense in which you will choose if it's physical, mechanical, or chemical. So those are the separates only content for plant diseases and defense. So this is all of the content you need to know for communicable diseases, including the separates only content. Remember that this is an overview, so you will need to study this in a lot more detail to make sure that you've actually got all of the specific things that you need to and content that you need to know.